Thank you so much. Thank you for the kind introduction. I'm pleased to be here. I'm extremely thrilled to be giving this lecture in the Bechtel Auditorium. This is a place where I had so many memories. I don't want to disclose. It's really a pleasure to be here and to be talking to you about a little bit about my research. It's extremely nice to be talking after Rana and after the senior year projects. The students that we saw, they were extremely nice. I'm very thrilled to be talking after them. My title for today is Make a Wish, and I don't want to say it comes true. It most likely won't, but at least it keeps you motivated and something else will happen. And that's why the title of my talk today is Make a Wish. And mainly it's related to coins falling in water. But I want first to tell you that I live in Los Angeles. This is a picture of Los Angeles. My son chose this picture. He said, I should show this one to you. And I don't live in downtown Los Angeles. I work in downtown Los Angeles. I live closer to the mountains, but not so far high up. My path from AUB to USC was not without serendipity. I was applying to graduate school while at AUB, and I was applying to MIT. It was at a time where we had to ask for the application to be mailed to us by snail mail. And I had received the application from MIT, and I was waiting for an application from Stanford to arrive when a friend of mine said, I got 2 applications from Berkeley by mistake. Would you like to apply to Berkeley? And I said, OK, sure, I'll apply to Berkeley. And that's how I ended up in Berkeley. It's not, it was serendipity, as I said. And then again, my path from Berkeley to Caltech had also some other detour. I was finishing my PhD, and I was really struggling, deciding between 2 opportunities of going to Johns Hopkins to work with Roger Ghanem, another graduate from USC, from AUB, who is a big name in uncertainty quantification and mechanics, or going to Paris 6 to work with Gérard Mogin at that time. And I wasn't able to decide where to go. And then I met a friend by coincidence in a coffee shop, and he said, what would be your ideal choice, your ideal situation? What would be the best thing that could happen to you? And I said, it would be going to Caltech to work with Jerry Marsden. And he said, have you written to him? And I said, no. And what's surprising about this is that I really never asked myself what would be the ideal place I wanted to be in. And just having someone from outside asking the question, just what would be the ideal situation? And then I acted on it. And that's how I ended up being at Caltech. I wrote to Jerry Marsden the same evening, and he replied the next morning. So as I just said, life is filled with serendipity, but again, that's how we end up where we are. And right now I am at USC. I've been there for 8 years almost, or maybe a little bit over 8 years. I want just to make a little correction on what you said, Kamel. The NSF Career Award, I received it in 2007, and then the KaprieIian Fellowship in 2011. They are 2 different fellowships. So I want to take you on a tour of my lab. It is mainly an academic talk that I'm giving. It's going to be a light academic talk. I'm not going to give you a lot of details, but I want to take you a little bit for a tour in my lab. I run the biodynamics lab at USC. I have now a healthy group of students and postdocs. I have 2 postdocs, Yang and Lionel. I have 4 graduate students, Han Liang, Sarine, Alan, and Yang Yang. Sarine is a graduate of AUB. She is now co-advised with a colleague of mine in biomedical engineering. She's working on neuromuscle control. And I'm not going to mention too much about what she is doing, but I'm going to highlight a little bit of the work of the others. And here are the 2 undergraduate students in the lab, Scott and Jeff. And the topics that we cover in my lab that we are interested in, they are mainly inspired by biofluid mechanics. I work on problems in biolocomotion. I did talk about this back in 2005 here. I'm interested in problems in collective behavior. For example, when this talk ends, we all have to leave this room, and there will be some sort of collective behavior happening with all of us trying, not knowing the status of the whole group, but still we do local adjustment, and there will be a global emergent behavior that takes place. So that happens with all animals at all scales, from the micro to the macro. And it's a very interesting and fascinating phenomenon, both physically but also mathematically. The structure of the equations that we need are different from our experience with normal partial differential equations that are forced at the boundaries. These are equations that are forced from within the system. The drive happens at the individual level. And also I'm going to talk about some biological flows. I wonder if is it clear? Do we need to dim the lights a little bit? Thank you. Okay, so I'm just going to tell you a little bit that thank you. So my approach to all of the work that I do is really to basically facilitate an integrated approach for both research, education, and outreach. And that's the way I try to run my group and my career in a way. So in my research, as I showed you, I had graduates, undergraduates, postdocs working together. The undergraduates have their own projects. I don't use them as support for the graduate students. They have their own projects. They do their own work. In teaching, I really believe in interactive teaching. I have my students come to the board and teach the other students. We exchange places. I sit in the student place. The students explain some stuff, etc. And I also believe in project-based education. My students this semester are working on implementing the game of life, Cohen's game of life. If you don't know it, go to YouTube and just put game of life. And it's a fascinating mathematical model that is capable of producing life and eliminating life in a very unpredictable way, starting from very simple rules of what is life and what is death. So they are right now working on this project, and they have to present it next week. So I hope they will do well. And also, we do a lot of outreach. And here, we don't really go ourselves and talk to schools. We do that through working with Mr. Larry Lim at USC, who is responsible for lots of outreach with local high schools in Los Angeles, mainly in underprivileged areas, where he invites those students to come to USC. We talk to them. Here are some pictures, just so that you see that my students are preparing some experiments for them. Mainly, we call this workshop Funny Fluids because we were having them experiment with viscoelastic fluids and with shear thinning and shear thickening by just dealing with substance that has starch in it and experimenting with that. And we also do some work with the Cabrillo Aquarium. The Cabrillo Aquarium in San Pedro is basically a teaching aquarium where they bring in middle school and high school students, and they work on testing some hypotheses. They teach them in the basics of scientific methods, and they spend the semester learning on formulating a question that is testable and then verifying whether the hypothesis is valid or not valid. And those 10 years old and 15 years old are amazing when they give their presentations. They stand there with a lot of confidence and they give extremely clear presentations. So I am really thrilled about my interaction with the Cabrillo Aquarium. This project is done by, under the supervision of Dr. Kirsten Darrow. She's doing fantastic with those kids. It's unbelievable. So going back to the work in my lab, we do motion in a fluid, and whenever we talk about motion in a fluid, basically we can classify this, whether we have inertial effects are dominant or viscous effects are dominant. So what do I mean by that? If you have a large fish, it has weight, it has mass, it has inertia, but not only that, it has muscles, it can flap against the fluid, it can push large amount of fluid with its motion, and that fluid has inertia. So that means that we have to account for the effect of inertia of the fluid as well. And that mechanism for moving is completely different from how little bacteria swim. Little bacteria have almost no mass, and they cannot push fluid the way that big fish do. So they use viscosity. It's a friction generated motion. They use viscosity and anisotropy in their movements in order to propel themselves in a viscous environment. And of course, we have plenty of organisms that live in between. For this talk today, I'm going to mainly shift back and forth between the two extremes, where inertia is dominant or viscosity is dominant. And of course, these works are the reason we look at this. We are interested in autonomous underwater vehicles that are efficient and stealthy and don't leave a big wake signature. And that's the main motivation for looking at these type of problems. And here there are plenty of biomedical and technological applications. Biomedical applications because we'd like to understand bacteria in order to prevent infectious diseases, and technological applications because there are applications in terms of biofuel reactors. And if we understand how to control bacteria, then maybe this could become a reality in the near future. So these are the main motivations for considering this type of work. And now I want to tell you about the scientific method that we use in order to solve these type of problems. And I thought the best way to illustrate to you the scientific method is to show you this drawing of bulls by Picasso. So here you see in the first sketch, you see a more or less faithful sketch of a bull. You see some details on the horns, details on the tail, the muscles, the legs. And then Picasso, he starts reducing the bull all the way to the last frame, where we see the bull is reduced to its main essence now. You have the horns, you have the tail, you have the legs, you have the genitals, you have the body of the bull. You remove any element of that, it's no longer a bull. You remove the horns, it's no longer a bull. So I think this is a great depiction of reducing something to its bare essence. And that's also something we try to do when we are formulating models of the physics. So you have, of course, I mean, researchers come in different flavors, the same as everything in life. Some people prefer to include all the details in a model, or you could try to look for models that are accounting for the main essence of the physics that's happening, and that's the style that I like. And I'm going to show you some of that in this presentation today. So this is a movie that I showed about 8 years ago when I came here, and that's a model of a fish. So in the first picture, in the upper row, you see the movement of a real fish. The flow field is constructed from particle image velocimetry. This is an actual flow field, and you see that this fish is pushing against the fluid. It's trying to push its way through the fluid. And the model that I worked on idealized this fish into 3 links. And now we have 2 angles that are deforming. So we are prescribing the deformation of these 2 angles. And we ask whether, if we put this model in a fluid medium, whether this would swim. And indeed, it does swim. And this is really a very minimal model. If we have only 1 joint instead of 2 joints, the fish will not swim. If we remove the fluid, this object will not move anywhere. It will rotate in place. It will become like a satellite, but it will not swim. So this is the bare minimum that is capturing the interaction of shape deformation with the fluid and how it's producing a motion. And we see that it actually produces a motion that's controllable. You can move, and you can also turn. So it's a completely controllable motion. Now I want to switch gears and go to the low Reynolds number, the viscosity-driven motion. And here I want to tell you a little bit that bacteria, when they are happy and there's plenty of food, they are in this free swimming mode where they have very high flagellar activity. And then when there is some stress on them from the environment, they go into a different mode, which we call the biofilm formation. There is a change in the gene expression that initiates this biofilm formation. This is a big problem in the medical industry. If you go to the medical school, probably you'll hear about it, that we don't want to have those biofilms forming. This is also the same thing that's on your teeth and my teeth right now. This is what you hear when you go to the dentist to clean your teeth. You hear, that's what they are trying to remove. So it's a big deal, the formation of those biofilm, and usually they are accompanied by a change in the flagellar activity of the bacterium. When the bacterium is happy and plenty of food, its flagellum is beating vigorously. When there is a stress in the environment, the flagellum is not beating as much, and then it switches into a state of biofilm formation. And here I'm showing a movie by a group of Harry Sweeney at Texas, where you see that when the bacteria are happy and free, they are basically, they form these big, large clusters that are at a much larger scale than the size of the individual bacterium. And this is what we see in here. So what we asked in my group is, could there be a role for the hydrodynamics in triggering the transition from free swimming to biofilm formation? And what we found is that indeed, in this model that we have here, we account for the flagellar activity, and we see that when we have high flagellar activity, the bacteria are happy and swirling and exhibiting this turbulent-like behavior, which we see here on your left. And then when the flagellar activity decreases, the bacteria start to cluster, which you see there in the aggregation. And this could be a mechanism that, I mean, we have to verify that, but it could be a mechanism that triggers the gene expression to change and triggers the switch to biofilm formation. So that's what I want to say about this model. I mean, this model is, maybe we could have a simpler bacterium, a simpler model of a bacterium, but we wouldn't be able to show this transition from swirling to aggregation unless we had accounted for the flagellar activity. And I think that we are the first group to account for the flagellar activity in these type of models. So I want to tell you now that bacteria, when it comes to us, they are almost always interacting with ciliated epithelia. So we all have cilia in our esophagus and the airways and our lungs, and these cilia have a very important function. These cilia are little hair-like structures. They are beating together, and they have a function to generate mucus and to generate fluid currents to move away this mucus that traps all the harmful particles that try to enter our lungs. So it has a clearing function. It tries to clear our lungs and keep them from having any harmful particles or bacteria entering into our lungs. So the point here is that bacteria interact with the currents that are generated by these cilia, and that there has been a lot of work recently on understanding behavior of cilia and cilia-generated flows, and also behavior of bacteria. But what we are trying to do now is trying to understand how cilia-generated flows help in generating a barrier between the bacteria and between the harmful bacteria and our ciliated epithelia, and while allowing for beneficial bacteria to exist in us, basically. Because we recently discovered that every one of us, you and me, and every one of us, not we in the sense, not me, but the scientific community recently discovered that we are all made of more bacterial cells than our own human cells. Every one of us has 90% more bacterial cells than human cells. And they are doing something. They must have a function. So only in the airway, there are about 100 different bacterial species that coexist at a density of about 2,000 cells per centimeter square. So they are inhabiting our lungs happily, but then when there is a harmful bacterium, then there is something that happens to try to expel it. And if that healthy process does not happen, then we are susceptible to disease. And that's what we are trying to understand in this project. And here I'm showing you basically the action of a single cilium. As I said, the cilium, you should think about it like a thin little hair. It's about 20 micrometers long, or it's between 5 and 20 micrometers long. And it forms a carpet, basically. And the cilia are not beating synchronously, but they are beating metachronally, that they generate a metachronal wave. And here I have a movie from my collaborator, Yana Novrot, on the cilia beating, but maybe the best way to tell you about what is the metachronal wave, I mean, you could kind of see a metachronal wave happening, and you see also how particles get deflected away from the ciliated field, but maybe I could explain to you the, let's see how. Maybe I can explain to you the metachronal wave by showing you this animation or this movie. Let's see. So this is a metachronal wave in a football stadium. It has a lot of effect when I show it in the US because football is so big. This is American football. And you see that everyone is doing the same thing, but everyone is standing up and sitting down, but they are doing it with a little phase difference. And that's what produces the metachronal wave. Do you know which movie is that? No, not the wave. Anyone wants to guess? It's from the 80s. What movie? It's from the 80s, should I? It's a very famous movie. Right, When Harry Met Sally. Marwan, you're in. So we have made this model where we are looking at a carpet of cilia. We're prescribing the motion of the cilia. We're taking the individual kinematics from some actual experimental work. And what we have found, in preliminary studies, we have two main results so far. One is that the cilia have dual function in the sense that they transport the fluid above the cilia tips, but they mix the fluid below the cilia tip. We're not the first ones to say that. There has been evidence, experimental evidence, suggesting that this is the case. But we are the first ones to show that the same cilia beating parameters that optimize transport also optimize mixing, which is not intuitive if you think about it. Why should the parameters that optimize transport above the cilia tip also optimize mixing below the cilia tip? And we also have a second result that is by comparing experimental beating kinematics, we found, and by comparing them in terms of basically different hydrodynamic metrics in terms of hydrodynamic efficiency, hydrodynamic forces and torques applied on those cilia, we have reasons to believe that cilia don't beat in a way that's hydrodynamically optimal. So there must be something else. There must be another biological function, whether it is, because cilia often serves two functions of sensing as well as generating flows. So there is something else that is dictating the beating of the cilia, and not just hydrodynamic optimality. And these models, as I show on the, you can see I'm showing the whole series of the bull, I mean, we can call it a bullometer, like a thermometer, and we see where our model is on this bullometer. So this model is probably with more details in it. Maybe we could have generated more or less similar qualitative results with simpler models. So here we are moving the scale up on the bullometer, if you like. Now I couldn't resist showing you this. I really couldn't resist because the story is so beautiful that my collaborator, my biology collaborator that works on cilia, that give us the data, the cilia that she works on, they don't come from human. The human cilia are very difficult to access. So she works on a model system. That's the model system that she uses is a squid. It's an invertebrate. It's a squid system where the story of this squid is really fascinating. This squid lives in shallow water, and its skin is mainly transparent, and it goes hunting at night. And so the moonlight goes through its skin, and mostly it doesn't cast a shadow except at the location of the ink sac, because every squid has an ink sac in its gut to defend itself. And that ink sac casts a shadow when the squid tries to go hunting at night. So to camouflage the ink sac, there is something called a light organ that when the squid, when it's first born, it has nothing in its light organ. But its job in the first hours of its life is to acquire a symbiont bacterium, a bacterium from the ambient fluid. That's a bioluminescent bacterium. And once it gets in its light organ, it uses it basically as a flashlight when it goes hunting at night. And it can basically control the amount of light that it wants this light organ to shed. So this is the system that we use as our model system. And here I show some images from my collaborator, Margaret McFaul-Nye, where she shows basically where the light organ is and how we have those ciliated horns and the pores where this bacterium should get to. So that's an example of a system that actually allows certain bacterium to come in and rejects everything else from every other bacteria or particle from the system. And we are trying now to understand how the cilia generated currents. So you see vortices. These are streamlines based on experiments of Jana Novrod. And you see that the streamlines look more like vertical streamlines. At this scale, by the way, I didn't tell you, this is about a few micrometers. I think you see the scale over there. We have 20 micrometers long. So this is also a viscosity-driven system. And we are trying to understand how these vortices, the vortex-like structures, help in sieving out particles and allowing only certain particles to get into the light organ. Now, I have a question, another question for you. Marwan, you're not supposed to answer. Where is this on the bolometer scale, this model? It's no longer a bull at all. We are interested in what's happening in the lungs, but we now are looking at a completely different system, which is the squid system. So we replaced the bull by a pigeon, if you like, because it's possible now to track. This is a more tractable model. It's possible to do experiments with that. And that's a technique that's often used in science, in scientific research, that you take a system that is not accessible or not tractable, and you replace it with another system that is tractable and accessible. And it could be both experimental or modeling, on the modeling end. So I'm going to show you another system. Now we are going back to the high Reynolds number. I'm going to show you another system where we replace the bull with a pigeon. This is the problem of flapping flight. You know, flapping flight is not an easy problem to solve. And this experiment, this really ingenious experiment by Jun Zhang and Lee Frostroff and Steve Childress at Courant Institute in New York, where they said, OK, we don't want to now flap the wings. We're going to oscillate the fluid. And we're going to put an object in an oscillatory fluid. Now, the fluid is oscillating in a completely symmetric way. There's nothing that's breaking the symmetry. The fluid is oscillating up and down by the same amount. But the only thing that is asymmetric here is the shape of this object that they put in this oscillatory fluid. If this were a completely flat plate, this object would basically, there would be no symmetry breaking mechanism in the vertical direction. But we have gravity is applied in the vertical direction, so you want something to break the symmetry to keep this object aloft. And that is basically the geometry that is breaking the symmetry. So here, what you're seeing here, you're seeing a pyramid-like structure that is oscillating in a fluid purely passively. There's no motor here, nothing, purely passively, and it's stable, purely passively. So the fact that it's hovering is not very surprising. Maybe you could reason it one way. But the fact that it's stable, that it's not tipping over, it's really surprising. And my student, Yang, she has been working on developing a model, a fluid structure interaction model, where she has a hat, basically, falling in an ideal fluid. The fluid model, the vorticity, that the vortex model is a vortex sheet model. She drops that in a still fluid with no oscillations. You see it just falls, and it falls really a long way. She drops that in an oscillatory fluid, and what you see here at the beginning is the formation of those dipoles here. This is the dipoles that I want to show you. You see a dipole that's shooting downward, and this dipole shooting downward, it produces a force that keeps this hat floating. And the same mechanism, this dipole shedding, is also responsible for stabilizing this hat, because when the hat tries to tip in one way, then the vortex shed, or the dipole that's being shed, produces a torque that restores it to its vertical proper position. So I know that Matthias is going to keep me on time. I have German accuracy here, German precision. So I'm going to finish with a problem on coins falling in water. That's what I promised you at the beginning. And the reason why we look at that, this is again a problem where we take the bull and we replace it. We take a proxy of the bull, which is the pigeon. We are basically interested in understanding unpowered flight. For example, if you have a spaceship that's trying to enter re-entry vehicles in any, to any atmosphere, to any planet, you cannot completely control its descent trajectory for obvious and non-obvious reasons. It's too much control to control the descent trajectory completely. So there is some, it is basically working under gravitational field and also under some aerodynamic forces. And of course there is some control, but it's not a complete control in this case. But other examples are the case of gliding animals, gliding snakes and gliding lizards in the rainforest. They can glide really long distance, completely unpowered. It's an unpowered flight. And understanding their gliding motion could have some technological, could be translated into some technological robotic vehicles. But also understanding the dispersal of seeds could have impact on understanding ecology, the plant ecology, and forestry, etc. So we replace this problem by the problem of dropping a coin, and this is now, I think, the hand of Scott, the undergraduate student in my lab, trying to drop a coin in the tank that we have. And you see that you have a completely non-trivial trajectory, a descent trajectory. And this is only one mode. I'm going to show you other modes of descent trajectories. So just to give you some background, if you take a coin, any one of us takes a coin and you flip it, this coin is subject to Newton's laws of motion. It's subject to mechanics, yes? Mechanics is not probabilistic. Mechanics is deterministic. We all learned that. So where does the fact that the coin could fall head or tail, where does this uncertainty come from? It comes from initial conditions. The uncertainty is in the initial conditions. It's not in the mechanics itself. And it has been established recently by Diaconis and other researchers that really coin flips are not fair. They are all biased, except when you go to a place in the space of initial conditions where you give it extremely high velocity, extremely high angular and translational velocity. Then it becomes more and more fair. But as long as you are in a different space, in the phase space, then the coin is unfair. And we tried this experiment with my 6-year-old son. He flipped coins repeatedly 10 times. I think he got 10 heads. I tried it. I think I got maybe a little bit better. And his father tried it, and his was mainly fair because he was giving it a very vigorous flip in the beginning. So just, I mean, it's not a proof, but it is just an evidence that, indeed, I think coin flips in space and air are not fair. So now what happens when they are in a fluid environment? So as I said, there is rich dynamics that's happening because of the interaction with the fluid, because of the vortices that are here. I'm showing you a PIV image that is taken by Scott, but also with the help of Lionel, our postdoc in the lab. So you see there is a non-trivial interaction of the vortices that are trailing behind the coin. And it has been known already from almost 15 years ago or more that we have several regimes of falling. When you fall in a fluid medium, you could switch from steady falling. So if the object is very heavy, it's just going to fall steadily if the coin is very heavy. But if the coin is, so if the moment of inertia versus the Reynolds number in the fluid is varied, then you could get a fluttering regime. That's the regime I showed you, where the coin was fluttering from side to side. Or you could get chaotic falling, or you could get tumbling motion. The tumbling motion is extremely interesting. If I had a piece of paper, maybe I could show you an example of tumbling. Maybe this will tumble, I don't know. So here it didn't tumble. I don't know how to categorize this motion. I probably should make it a little bit straight. No, it doesn't want to tumble. It just wants to flutter, so I'm sorry. No tumbling motion now, but you could get this tumbling motion. And the question that we asked is, okay, so you have motion in those different regimes. How does that translate to landing sites? So where would you land? If a coin is dropped in a fluid medium, where would that land? And the answer is, of course, probabilistic, because it depends on your initial conditions. So we tried to, the uncertainty in your initial conditions. So we tried to minimize the uncertainty in the initial conditions. So we're always dropping the coin with the same initial conditions. Yes? This is the work of my undergraduate student, Luke Heisinger. He now graduated and has a job. But he was trying to maintain the same initial conditions, dropping the coin repeatedly, and just recording the landing sites. And what you see here is that the landing sites are extremely different depending whether you are falling in steady or in fluttering mode or in chaotic or in tumbling mode. So if we now construct probability distribution functions, what we see that when you're falling in a steady mode, you're always gonna land under the point where the coin is dropped. When you're falling in a fluttering mode, there is a dip here. There's a dip around 0, which tells us that the probability of landing under the place where the coin was dropped is really not very high because there is this big dip. And the same in the chaotic mode, we get very broad probability distribution function. And here, what I'm showing you here is basically that when in the tumbling mode, the probability of falling right under the place where you've been dropped is completely 0. So this has been exploited by people in advertisement or in entertainment business in Las Vegas and other places. So the idea is that you have some machines where you go and they promise you if you can drop a coin right in a given spot, then you're gonna win something. But they know that they can bias the coin, the parameters of the coin, to minimize the chances of the coin being dropped or landing in a certain space. So now the question is, can we, if we want a desired landing site, can we design a coin or a lizard or whatever it is that takes us to that landing site? That's the inverse problem. That's a much more difficult problem. And I also wanted to report to you, since I started by talking about fair coins and flipping basically head or tail coins, I wanted to report to you that for these experiments that we did, when the coin is being dropped vertically in steady or in fluttering mode, the probability of landing heads is always 1. We always start heads up. The probability of landing heads is always 1. When the coin is in the chaotic regime, then the fluid medium acts like basically a randomization device. The probability of landing heads now becomes almost half. And when we are in a tumbling regime, then you can bias the probability the way you want by controlling the height, because when you are in the tumbling regime, you are in this periodic tumbling motion, and you can bias the probability depending on the height from which you drop the coin. So I want, yes.
What about the two surfaces of the coin? Don't they have any?
Good question. The question is, what are the two surfaces of the coin? These are the coins that we used. These are coins that we manufactured, so they were completely. But now we are adding roughness effects. That's a different question over there. Actually, we are also adding a hole in the coin, and we see that it stabilizes itself. So if you are interested in any of the stuff that I mentioned, or you're interested in knowing some of the stuff that I did not mention, please contact me or any one of my group. Please don't call, email. I rarely answer phones that are in my office, but I always answer email. And if for any reason I did not answer the email immediately, it's probably because I am trying to chase behind those two little devils, Stefan and Ziad, or I am at the gym and I want my one. I told him I have been taking kickboxing lessons. He was very impressed.
Please practice on him.
Yes, I would be happy to do that. So all the courses that I took with him. So I just want to leave you with two quotes. I want to leave you with two quotes from my kickboxing instructor. She says, pass the ball, which basically when we are exercising, the ball becomes extremely heavy. You have to pass it very quickly, otherwise you're gonna die. And she's serious, she's very serious. So I learned that, and I try to apply it now at work. basically, I try to get rid of whatever I need to get rid of. I just pass it back to my students or to my colleagues, or whatever I have to pass, whatever heavy thing I have on my desk, I try to pass it immediately. And that's my favorite quote since I've been taking those classes, pass the ball. And then her other quote that she keeps saying, other expression that she's very serious about. Of course it's hard, you have to do it. I mean, of course it's hard, you know. So I want to thank you very much for listening to me. I'm happy to take questions if you have any.
Your outreach, was that the career award? The outreach activity.
A result?
Is that how it started?
It started, yes, during my career. Yes.
Now, I have one more thing to say on the bacteria.
Yes.
When they flocculate or they go on revival,
yes.
Sometimes it's a more efficient way of using the food effectively because of the redox conditions.
Right. That's correct. So whenever there's stress in the environment and the sense of learning is increasing, we try to enter the environment. But to enter the environment, they start secreting and the others start right. So they have to have some applications on this. And what we are interested in is really the transition from this to agriculture.
Some very nice work was done on this on submarines. Because submarines, they have a biofilm that attaches on them, and then the biofilm will release some products. And you can break hack submarines sometimes by looking for the products. And that's how the research was started initially with the Navy.
Eva, in the feeding system, now if you go to the limit where you actually have zero friction along the length of the body and you have friction sideways, you have a purely kinetic system. In that case, you can actually parallel park, means you move sideways.
That's exactly what we do.
Right, but if I go to your example where you don't have that ideal case, can fish still go sideways somehow?
I do have that.
You don't need to be truly kinetic to go sideways. Parallel parks.
This is schematic in the sense that you can use your equations first order. It's not schematic in the sense that those first order equations have purely. So your first order equations are now schematic equations. They have counter.
The kinetic in my head is when you freeze your body, you stay with this.
That's exactly what happens. That's exactly what happens. Stop actuating, stop moving.
Anyone?
Your model solid fluid interaction to the fluid, you have the material size of solid part decreases in size. Is there a threshold volume after which the physical mechanism changes from inertia to viscosity? I mean, how do you determine the threshold for the mechanism to go from inertia to viscosity?
So you look at the Reynolds number, and if the Reynolds number is intermediate, then you have to account for it. If the Reynolds number is extremely high, then maybe you can, of course you lose something by making those assumptions. That viscosity is always there, but if the Reynolds number is extremely high, then viscosity is maybe small, but it's still there. Bacteria, it's really clear that the Reynolds number is so small. It's of the order of 0.0005 or something. That you basically assume that you are in completely Stokes flow. And that's so it's clear.
Does the equation, is it valid at least?
Right.
Are they?
Yes, they are valid. Stokes equations are basically saying you get rid of the
balance of viscosity.
Thank you.